diabetes, we call that. Whether we're calling mellitus or insipidus, two different forms of this copious urine formation. And then a chronic problem, renal failure, and how renal failure is treated. Finally, at the end of today, I'm barely going to get to the way that different animals control their osmoregulatory environment. And that'll lead us into the lab, where we're doing the fiddler crab lab this week. All right. So really quick, again, we're looking in the nephron, and all of these figures I'm about to show you from your textbook, they are looking at the lumen. Okay, when I say the word lumen, what does the word lumen mean? The inside, right? The lumen is the inside of the nephron is what we're speaking of here. So the fluid inside here is the lumen. We call it the luminal side. Here would be a cell. Just make sure you know where we are here. This is a cell that you're looking at right there. This is this cell. This is the luminal side. This is the peritubular capillary that's on the outside. And this surface right here is called the basolateral surface. So if you want to think about it, there are two different epithelial sides of this cell. The luminal epithelium and the basolateral epithelium. Make sense? Okay. Now, let's go into what happens along these different sides of the epithelial cell. The first way that substances are transported is passively, using no energy at all. Passive transport. Great example is glomerular filtration. We talked about that earlier in the semester. The last class, I talked about glomerular filtration rate. This glomerular filtration doesn't take any ATP, doesn't take any energy. It just happens because of blood pressure. So you can see right here, filtration into the lumen is passive. No active process at all. When we get to active transportation of substances, we've really got two options. We have primary active transport and secondary active transport. So let me spend just a few minutes to talk about primary active transport. By the way, if you need a refresher on active and passive transport, it's in chapter 4 of your textbook. We're intro chem information, so I'm going to assume you all know active and passive transport. Did you survive?
Yeah.
Okay, so let's look then at primary active transport. On the basolateral epithelial side, everybody good with that? What I'm talking about? Basolateral epithelial side, we have sodium, potassium, ATPase pumps. It is the, okay, so we've got one epithelial cell. See, right here? This nephron has only, it's simple squamous epithelium. It's one cell. It has two sides to that cell. It's just the outside, if you will, instead of the inside. The basolateral is the outside of the cell, if you're talking juxtaposed to the peritubular capillary. On the basolateral side, we have sodium, potassium, ATPase pumps. Now, every cell in your body has sodium, potassium, ATPase pumps, but these are only on the basolateral side. So, let's think about that. If we got sodium, potassium, ATPase that are pumping sodium out and potassium in, 3 sodiums come out, 2 potassiums go in. This is going to literally lead to reabsorption of the sodium. Think about that real quick. Last class, we talked about reabsorption and secretion. Luke Bryan concert that you're still conscious. What do these two words mean? We're talking about the kidney and the nephron. Saving stuff, right? Not letting it in your urine. Secretion is?
Secreting it back into the urinal.
Secreting it back into that nephron. Okay. So, what we've got here is a primary reabsorption of sodium because it's only on the basolateral side. Pretty simple. We also have examples of secondary active transport. Now, there are two kinds of secondary active transport. There's co-transport, and there's counter-transport. This is figure 28.3 in your textbook. Let me start off with the top. Forget the bottom real quick. Now, just like I mentioned, we are always actively pumping sodium on the basolateral side. Sodium is leaving. What would happen if we set up a bridge on the luminal side that every time a sodium molecule went in, it grabbed onto a glucose molecule and brought it with it? Now, this doesn't take ATP. This passively happens. But follow me real quick. We have a protein bridge, a transmembrane protein, on the luminal epithelial side that conformationally is structured. So only when we go have sodium go in, we'll always have one glucose molecule. One sodium, one glucose. One sodium, one glucose. Follow it real quick. I turn this pump on. By osmosis, sodium flows in here. But there's so much sodium now that's needed to maintain osmolar homeostasis that this sodium glucose transporter protein split. Sodium glucose transporter protein always brings glucose and it reabsorbs it. After glucose gets here, we have a glucose transporter molecule that's on the basolateral side. That is called co-transport. Why? It's going with sodium every time. Now, here's the trick question. Is this using ATP? I know. How's it using ATP?
Sodium.
Because it relies on the ATP of the sodium potassium ATPase bond. It's an indirect. It's a secondary active transport process using co-transport. That happens on the proximal convoluted tubule. I discussed that last class. We reabsorb 100% of the glucose into our bloodstream in that proximal convoluted tubule. And the way that that happens is through secondary active transport. Let's talk about the opposite, counter-transport. Now to understand that, let's go down here to the bottom. Again, all over this nephron, we have sodium, potassium, ATPase pumps that are pumping sodium out and potassium in. What if we expressed on the luminal side, instead of a split, a sodium-hydrogen exchange pump? In this case, the protein conformation is so that whenever sodium goes in passively, no ATP is used here, a hydrogen ion leaves. It's like a gate that only works if you have both those players there. Anytime sodium goes in, hydrogen is secreted. Why am I saying hydrogen is secreted? Where's hydrogen ending up? It's in the nephron now, and it's going to go out in the urine. This is not a primary active transport process, it's a secondary active transport process. So, let's consider the miracle of this nephron really quick. What do you think is the real difference between an epithelial cell that is right here from one that is, say, right here? What's the difference in an epithelial cell? The differential expression of proteins on that epithelial membrane. Bam. Those are the ways that substances are moved in and out of the nephron. Okay. Now, I've been talking about these proteins. Let me recognize that there's a transport maximum for proteins. If we look at, let's just take glucose because it's the simplest. If we look at the concentration of glucose in the urine, you survive that?
That was not a happy face you
No,
showed.
not a happy face. If we look at the concentration of glucose in the urine, and we plot that against the concentration of glucose in the plasma, that'll be in milligrams per deciliter. You recall that homeostasis for glucose in your plasma is about 100. As long as glucose is in a normal range, there's going to be no glucose in your urine. But after you exceed a normal physiological range, you will have some in your urine. And the reason is because there's a transport maximum that is dictated based on the number of proteins that are either co- or counter-transported. In other words, right here, what is the reason that, okay, now let's review. We had lots of people just come in, they have no idea what I'm talking about now. I'm talking Greek. Somebody explain to them really quick. How does glucose literally get reabsorbed?
Split.
Split?
Split.
Now you're speaking Greek. What does that mean? What is split?
Sodium glucose transporter.
The sodium glucose transporter protein. Sodium glucose transporter protein. What if we only have one sodium glucose transporter protein?
You're going to lose a lot of glucose.
You're going to not be able to reabsorb that glucose. What if you have, let's just pie in the sky, what if you have 150 glucose splits? What if you've got in your blood, though, 500 glucose molecules and 150 splits? You're going to have glucose in your urine.
Yeah.
That's what I mean when I say transport maximum. Anytime we have excessive of normal homeostasis, we're going to end up spilling whatever that molecule is.
What if you don't have enough sodium?
Okay, yeah. You die. Yeah, because
you just die. You die.
Now, the reason I would say sodium necessary for action potentials.
Okay.
So even before we see any osmoregulatory problems with sodium, we're going to have motor function problems, neurological problems. So you'd be dead before you'd see a problem with the sodium. Yeah.
It's the least of your worries if you're
That's why I salt my French fries.
That's why I put salt on everything.
Okay, so we've talked about the way that substances are moved into and out of the nephron. Let's turn the page now and talk about maintaining enough sodium in our blood. Maintaining enough ions in our blood. We call that osmolar homeostasis. Osmolarity in general refers to the concentration of solutes in a fluid. Solutes. What does that mean? What's a solute?
Like things dissolved into solution.
Anything dissolved in the solution is a solute. Could be glucose. Here's the reason that we talk about sodium always, is because when you look at the concentration of sodium in your blood, it's way high compared to any other molecule in there. So the most contributing solute to your plasma is sodium. That's why I'm going to be talking about sodium a lot. There are 3 ways that we physiologically control osmolar homeostasis. Sodium concentrations. The first one is the ADH system. ADH is a hormone. It's made by your hypothalamus, and it's released by your posterior pituitary. It's anti-diuretic hormone. So what does ADH do? So here's your textbook figure again, figure 2910. It shows you that it's made up here in your hypothalamus. It's moved down to your posterior pituitary and released into your bloodstream. This is in response to high osmolarity, high solute concentration. Anytime you become dehydrated, for instance, your salt concentration goes up in your plasma. All of your solutes go up. That causes these cells in your hypothalamus, osmoreceptor cells, to shrink by osmosis. Right? Those cells lose water by osmosis. When those cells shrink, they stimulate the supraoptic neurons. These are neurons in a particular part of your hypothalamus. We call it supraoptic because it's right above the optic center. The supraoptic neurons then make ADH, which is released into your blood. ADH goes to your distal convoluted tubule and your collecting duct. It targets those regions. And it targets those regions because that's where you have receptors. See?
Okay, this is just a question kind of related. Last week in our game, one of our girls was super dehydrated, like about to pass out, and they had her eat pickles. And I'm curious as to why, because those are high in sodium, so if your sodium is very high, then why would
Yeah. Good question. It will be to stop the sweating, right? To stop the loss. I would worry about her overheating, but yeah, it would be to any water loss.
just to track what you have?
Yeah, I think it's really high in electrolytes,
Okay.
so it would be to introduce electrolytes to hold any water in. The only worry I would have there is heat loss, because then without the evaporation, you can't have heat loss. But yeah, that would be the reason. Okay. let's look at the molecular mechanism of ADH's function. This is an epithelial cell. And guys, you guys that came in late, let me reset real quick for you. Whenever I'm throwing up these very confusing, very busy figures, we are looking at one cell of a nephron. Right now, I'm looking at a cell that has an AVP receptor. And here's where I can't stand your textbook because I've mentioned this before. Vasopressin is the same molecule as ADH. Vasopressin is structurally exactly the same. Arginine vasopressin and ADH are synonyms. So I wish they would put ADH there. They didn't. They put AVP. It's the same molecule though. AVP is equal to ADH. So just think about this as an ADH receptor. Whenever ADH binds to a receptor, what happens is aquaporin molecules are mobilized on the luminal side. Make sure you're following me. Here's your blood. You got ADH in it. It gets right here. Well, let's go here. It gets right here, beginning of the distal convoluted tubule. And what happens is, it binds, and on this side, it's going to cause these aquaporin molecules to go up. You guys remember those from Biology 115? Aquaporin. What does aquaporin do? It is bulk transport of water. It's basically, if you want to grab a bunch of water and either move it in by endocytosis, y'all remember endocytosis and exocytosis. The way you bring water in is through these aquaporins. Beautiful name. Aquapor. Water pour. They bring in a bunch of water. Follow me real quick. ADH binds, aquaporin, mobilized, bringing water out of the tubule, moving it back into your bloodstream. What did that just do to the volume of urine?
Decreased.
Decreased. Anti-diuretic hormone. Doesn't allow urine formation. Indeed, we can look at this another way. This is a different chapter in your book. Chapter 29. We could look at this in terms of movement of water, or we can look at it as concentration of the urine. So here we're looking at the concentration of the urine, either with low ADH or with high ADH. Now again, high ADH means water is leaving the filtrate, the urine, going back in the body, leading to a really concentrated 1200 milliosmol concentration. Without ADH, there's a lot of water. All right, let me bring it home and bring it real life for you. I have heard, although I haven't personally experienced, I've heard that alcohol consumption causes excessive urine formation. I have witnessed this. My friends, at a bar, friend, and we could do an experiment. We're not going to. We could do an experiment.
Volunteers.
We could do an experiment where you would literally measure, let's say, a pint glass of beer consumed 2 pints of urine produced.
The what?
Input 1, output 2. bizarre. This would be a fun lab, wouldn't it? We could sit.
Maybe that's a
5 pints of beer.
Or 10 pints. Everybody gets a graduated cylinder, has to go to the bathroom. Yeah. All right, make friends quick with that one. Yeah. Ethanol.
Conspiracy
Oh, we're with
ADH inhibitor, maybe? Called it.
Ethanol is a potent inhibitor of antidiuretic hormone. Ethanol shuts down your posterior pituitary. Sounds bad. It is.
No.
It's. I'm going to bring this up later in the semester. We're going to talk about another hormone. I'm just going to tease you with it right now, so you'll come back later in the semester. Later in the semester, when we talk about childbirth and labor and uterine contractions, the only other hormone your posterior pituitary makes is oxytocin, the contraction. And physicians in the olden days, when a woman went into preterm labor, they would prescribe her whiskey. And it stopped uterine contractions because it shuts down the posterior pituitary. Just like it shuts down ADH production. Okay, so the problems with excessive alcohol consumption are dehydration associated. The hangover it's a dehydration effect.
Wouldn't ethanol draw water from wouldn't it bring it into the luminal side and bring it into
there.
It's not there. It's actually detoxified. So by the
it's
liver,
in the liver, yeah.
before. But the metabolite that's detoxified is actually what's inhibiting the posterior pituitary. Make sense? Okay, so now you know the way that ADH can control volume of urine. I'm just barely going to mention the second way that osmolar homeostasis is maintained in your body, and that's with this thirst center. A part of your hypothalamus controls the desire to drink water. And anytime those supraoptic nuclei shrink, not only do they stimulate ADH production, they also stimulate thirst. So you introduce more water. The third way, that's pretty simple. The third way has to do with hormones. And I'm going to talk about two different hormones that control osmolarity. The first one is called angiotensin II. We've studied angiotensin II already. Angiotensin II, again, I'm showing you another piece of a nephron. Angiotensin II binds to receptors that stimulate both sodium-potassium ATPase and sodium hydrogen ion exchange. So what's this going to end up doing? Angiotensin II, yes, it's a vasoconstrictor. We talked about that before with blood vessels. It also causes reabsorption of sodium. You see how this would reabsorb sodium? I mean, sodium is leaving your tubule lumen, going back into your bloodstream. In other words, what influence is angiotensin II having on your osmolarity in your plasma? Up or down?
It's increasing the amount of water.
It's increasing the amount of solutes, so it's going up. Now, it's going to eventually have a negative feedback effect with water. The bottom line is, if you give somebody an injection of angiotensin II, their sodium reabsorption increases, spiking sodium concentrations up really quick. The opposite hormonal effect of that are a hormone family that we call natriuretic peptides. Natriuretic peptides. have only recently been discovered in about the last 10 years. They are made by cells all over your body. Anytime you see the suffix uretic, what do you think? Urine. Anytime you see the prefix na, sodium. There we go. Natriuretic peptides cause sodium to go in your urine. The opposite that we just had. Make sense? We still don't know much about them. We know that your brain makes a lot of this molecule, this hormone. We know that your heart does. And so anytime you look in the literature and you see a natriuretic peptide, it'll always have a prefix before it, like brain natriuretic peptide, BNP it's called. Or cardiac natriuretic peptide, CNP it's called. For instance, an article that was published just a couple weeks ago, Journal of Sports Medicine. Travel to high altitude is increasingly popular. People like going up where they should be going. With this comes an increased incidence of high altitude illness, and therefore an increased need to improve our strategy to prevent and diagnose these. In this review, we provide a summary of recent advances relevant to practitioners who may advising travelers to altitude. Although the Lake Louise score is now widely used as a diagnostic tool for acute mountain sickness, increasing evidence questions the validity of this and considering acute mountain sickness as a single condition. Biomarkers of this mountain condition, such as brain natriuretic peptide, BNP. Anytime you see BNP in somebody's plasma, it's a warning sign for mountain sickness, are likely correlated with pulmonary artery systolic pressure. Thus, okay, I'm going to stop right there and make sure you're following based on the last couple weeks of lecture. If you go up in elevation and your BNP goes up, what is BNP? Brain natriuretic peptide. What effect will that have on your blood pressure? Increased blood pressure. Why?
You're losing sodium.
What do you mean losing sodium?
Like it's being it's being not reabsorbed.
Okay.
It's being secreted back into the.
It's being secreted.
Yeah.
Yes, into the tubule. So what's that going to have on your blood pressure? Sodium is being spilled without enough water being able to follow it. The end result is excessive water in your circulatory system. So it's going to have a cardiovascular as well as a urinary output. Let's continue that mindset. Diseases associated with altered urine osmolarity. Okay. Following the same logic, I'm going to talk about the two most common diseases. They're both given the name diabetes. Diabetes is actually a Greek word for siphon. Water loss. And there are two forms of diabetes. We've already discussed diabetes mellitus. Mellitus is Latin for sugar or sweet. Sweet excessive urine. Insipidus is the word insipidus means mysterious, excessive urine. I want to follow through both of these diseases really quick and talk about how they can alter the osmolarity of your urine. Diabetes mellitus. Let's go back to exam 1 material. What is the problem with somebody with IDDM? No beta cells in the pancreas, therefore, no insulin, therefore, high blood glucose in the urine. It's been filtered, therefore, the urine, that glucose is going to be spilled because the glucose transporter pumps have been exceeded. The urine is going to be sweet. As that glucose is in the urine now, it's bringing by osmosis water with it. Everybody good with that? Water is following the glucose. individuals with diabetes mellitus are urinating about 15 liters a day. 15 liters. Insane. They're drinking tons of fluid and spilling all that fluid. Leads to cramping because they're spilling electrolytes. So the telltale signs for IDDM: excessive urine formation, cramping, glucose in the urine. Bam, diabetes mellitus. Let's contrast that with diabetes insipidus. Still 15 liters of urine, but it doesn't have any glucose in it. So what do you think could be the problem here? What's the hormone that we know that if you don't have it, you produce a ton of urine? ADH. The problem with diabetes insipidus is a reduced ADH. Head injury can cause this. Infection can cause this. Or sometimes it's a congenital condition where you basically just inherit the inability to express enough ADH. Like we know, diabetes mellitus is treated with insulin injections. Diabetes insipidus is treated with ADH. Bam. Healthy again. Okay, so those are the conditions associated with altered urine osmolarity. Let me transition to the third topic I'm going to talk about today, and that is when the kidneys start to fail. We can talk about kidney failure as either being acute or chronic. Which do you want?
You
Oh, either, but you got to pick one.
Oh, talk about or have?
Half.
Oh, here.
I want that long term. I want that long permanent one instead of the short term
Just keep
temporary.
drinking the alcohol.
Sorry, guys. Look, Brian. It happens.
He sold his kidneys.
These are the effects of alcohol and ADH.
I'm still here. Dang it.
We're going to talk about acute, short-term disruptions, and I'm going to throw up two tables here. I do not expect you to memorize the information on these tables. I just want you to look at them and understand them, okay? Very busy, awful.
What is this?
Different things that happen acutely. We've either got pre-renal or infrarenal kidney failure, either right before the kidney or inside the kidney. The pre-renal acute kidney failure can happen because of diarrhea, vomiting, excess fluid loss, heart problems, heart attacks, anesthesia effects, primarily on the nerves losing their function. Inside the kidney, we could have the tubule having epithelial injury that can be repaired through normal mitosis. Or we could have the vessels and glomerular injury that through angiogenesis are fixed. So these are acute problems. More problematic, I want to say, no doubt, is chronic kidney failure. CKD, you will see it referred to as. And again, there's lots of reasons for this. The biggest reasons are diabetes mellitus, obesity, and hypertension, all of which are in epidemic proportions in our population today. Information from the Center for Disease Control, the CDC. Looking at chronic kidney disease, in other words, there's no turning back. We've got a problem that can't be repaired through mitosis or angiogenesis. Individuals today in the U.S. older than 65 years, about 45% of the population has chronic kidney disease. As we, thank goodness, keep that's alive longer, we're going to see this escalating. And as we have more and more diabetics, people with hypertension, elevated cholesterol, these are going to contribute to chronic kidney failure. Okay, chronic kidney disease, bad problem. Everybody good with that? Let's get on the solution side. How do we fix this problem? Well, right now, the most common solution is dialysis. Technically called hemodialysis because we're using the blood and we're dialyzing it. Many of you may have done in intro biology or intro chem dialysis membrane experiment. Dialysis just means semi-permeable. You're able to move some substances across and save some substances. And dialysis, see right here in figure 32A, is where an individual is hooked up, their blood is pumped externally through a dialyzing fluid. This dialyzing fluid pumps the bad stuff out and concentrates the good stuff. I'll show you a list of the bad stuff and the good stuff in just a second, once you get the big picture. Dialysis is a life-changing treatment. 3 days a week, typically, a person that's on dialysis will go to a dialysis clinic. Each session is between 3 and 5 hours, sitting there on the dialysis machine. It's about $80,000 a year. Goodness gracious. Thank goodness right now insurance is absorbing some of that, but that's changing. Insurance companies can't afford this anymore. This is one of the reasons for the big problem. You can see this is a solution, but it's not a great solution. This solution, here's your dialyzing fluid. The fluid basically is going to try to save the good stuff. Somebody that has chronic kidney failure has what we call uremic plasma. What do you think's in that? Lots of urea. There's urea, really harmful, nitrogenous waste, toxic to our body. This dialyzing fluid basically has no urea. It's removing the urea. It's also introducing a bicarbonate ion, which we're going to talk about that a lot next class when we talk about pH balance. So what we're doing is we're saving the good, releasing the bad. Okay, so that's one solution, and you can see it's not a good solution. A better solution. Let's get a good kidney. Put them in there. Most people don't know when they do a kidney transplant, you don't like take the bad one out. It doesn't make much sense. You add another one. Individuals that have had kidney transplants have 3, 4 kidneys. The reason is because it all goes back to the nephron. A kidney, even in chronic kidney disease, you've had 1 million nephrons. Even if you've got 100,000 left, they're doing something. Why remove those? And why cause all the problems with snipping the renal artery and the renal vein and having, leave it in there. Add another good one. Kidney transplant, clearly a wonderful technique. In the U.S. last year, there were hundreds of thousands of kidney transplants. The limitation here is kidneys. If you are not right now on the organ donor list at the DMV, go do it. I mean, it makes no sense not to be an organ donor in my mind. If I die today, I hope every organ in my body goes to help somebody else. The rest of them burn me up and spread me in the woods or something. I mean, you know. But even if everybody was a kidney donor, we still wouldn't have enough. And we still wouldn't have good matches, tissue matches. We talked about that a couple of classes ago. What do we need to match? HLA antigens. We have to have an HLA antigen match. So we're all of a sudden down right now. And then we need to have immunosuppressants. Oh, frustrating. There's got to be a better solution. Don't get And indeed, welcome to the future of treating this problem.
Is this a TED Talk?
There we go. We're not going to watch all this.
Of course not.
We're going to watch just a little bit of it.
There's actually a major health crisis today in terms of the shortage of organs. The fact is that we're living longer. Medicine has done a much better job making us live longer, and the problem is, as we age, our organs tend to fail more. And so currently, there are not enough organs to go around. In fact, in the last 10 years, the number of patients requiring an organ has doubled, while at the same time, the actual number of transplants has barely gone up. So this is now a public health crisis. So that's where this field comes in, that we call the field of regenerative medicine. It really involves many different areas. You can use actually scaffolds, biomaterials.
The leading lab
They're
is
like
indeed right across the mountain in Wake Forest for
but
regenerative
specific
medicine.
materials you can actually implant in patients and they will do well and help you regenerate. Or we can use cells alone, either your very own cells or different stem cells, a printer, and implant it as you see here. That's all new bone that was implanted using
He's gone through
these
now.
techniques.
They're doing this with bladders. They're doing this with bones.
Another more advanced technology we're looking at right now, our next generation of technologies are more sophisticated printers. This particular printer we're designing now is actually one where we print right on the patient. So what you see here is, I know it sounds funny, but that's the way it works, because in reality, what you want to do is you actually want to have the patient on the bed with the wound, and you have a scanner, basically like a flatbed scanner. That's what you see here on the right side. You see a scanner technology that first scans the wound on the patient, and then it comes back with the print hits, actually printing the layers that you require on the patient
These are different cell
themselves.
types.
This is how it
Epithelial
actually
cells,
works. Here's a scanner going through,
connective tissue
scanning the
cells.
wound. Once it's scanned, sends information in layers, the correct layers of cells where they need to be. And now you're going to see here a demo of this actually being done
Currently being used in burn
in
victims.
a representative wound. And we actually do this with a gel so that you can lift the gel material. So once those cells are on the patient, they will stick where they need to be. And this is actually new technology still under development. We're also working on more sophisticated printers because in reality, our biggest challenge are the solid organs. I don't know if you realize this, but 90% of the patients on the transplant list are actually waiting for a kidney. Patients are dying every day because we don't have enough of those organs to go around. So this is more challenging. Large organ, vascular, a lot of blood vessel supply, a lot of cells present. So the strategy here is this is actually a CT scan, an X-ray, and we go layer by layer using computerized morphometric imaging analysis and 3D reconstruction to get right down to those patients' own kidneys. We then are able to actually image those, do a 360-degree rotation to actually analyze the kidney in its full volumetric
Everybody's kidney is slightly
characteristics.
different.
And we then are
Based on
able
your
to actually
morphometrics.
take this information and then scan this in a printing computerized form. So we go layer by layer through the organ, analyzing each layer as we go through the organ, and we then are able to send that information, as you see here, through the computer and actually design the organ for the patient. This actually shows the actual printer, and this actually shows that print.
What they would do here that's a little bit different is they would go into the patient, he went into this earlier talk, go into the patient and take a biopsy. Take the cells, dissipate them. Take the epithelial cells and grow them in medium. In other words, there's no problem with the HLA antigens because they are yours. This is, talk about personalized, these are your cells that are being grown in vitro, right? Different cell types, epithelial, connective, nervous, muscular, putting them all, and then getting a printer that's going to, those that you've grown using growth factors, is then going to squirt them in the right layering.
Printing. In fact, we actually have the printer right here. So while we've been talking today, we've actually, you can actually see the printer back here in the backstage, that's actually the actual printer right now, and that's been printing this kidney structure that you see here. It takes about 7 hours to print the kidneys. This is about 3 hours into it now. And Dr. Kang's going to walk on stage right now, and we're actually going to show you one of these kidneys that we
So
printed
these are different
earlier
cell
today.
types that. Funny, you can tell this guy really doesn't get to lab much here. Watch his lab clothes here. He can't get a lab glove on.
So these clothes are a little bit smaller, but here it is. You can actually see the kidneys that was printed earlier today.
Yeah.
Has a little bit of consistency to it. This is Dr. Kang, who's been working with us in this project, and we one part of our team. Thank you, Dr. Kang. Appreciate it. So this is actually a new generation. This is actually the printer that you see here on stage, and this is actually the new technologies we're working on now. In reality, you know, we now have a long history of doing.
What are you doing? You're still talking.
So, as I've mentioned countless times already this semester, the future of medicine is really STEM related. It's the merging of engineering with what we know about medical problems. No longer is our emphasis really drugs, a pharmaceutical agent. We know now we're going to have more better surgical procedures, better transplant procedures through STEM technology. Yep.
With the kidneys they're making, it's still just attach it and put 3 kidneys in there, right? They would
Yes, exactly. Same thing. You would still add a new one, and really, it's kidney transplants relatively easy because there's one artery, one vein. Plug it in, hook it up to the main, and you're done. It's not like the vascular supply is very difficult. Anybody else?
It said obviously there's still years away from using that, but have they tested it in a lot of patients?
Not kidneys. So bladders, definitely. Bladders are already in. They do different phases of. They finished phase 2 testing. So many people are walking around with printed bladders right now. Kidneys still a little bit away.
So are they scanning the damaged kidneys?
Yeah, but you can see what the problem is. Yeah.
They know how to correct that.
Yes. Yeah. What you saw in the image, those black regions, that's where you should have had nephrons in there. So it replaces, kind of like just a negative, any place where you have negative space, you put the nephrons back in there. Anybody else?
So are they looking at doing this with just like kidneys, bladders, etc., or are they looking at different organs? Like I know one of my sister's best friends has been on the lung transplant list forever, and she just got lung cancer.
Yeah. Kidneys are the number one emphasis right now because of chronic kidney disease, and its lungs are even more problematic because of the vascularization. Sure, every one of these does have a nephron, a capillary bed that goes to it, but the alveoli, every one of those has such. Theoretically, definitely. I think the first one that'll come on whole organ transplant will be kidneys. Next will be liver, mostly because of demand, and then others one day. Okay, let's look at osmoregulatory strategies of animals. The last thing I'm going to talk about today. When we talk about animals, we can talk about an animal either being an osmoregulator for an osmoconformer? Osmoregulators keep their internal osmolarity constant, no matter what happens to the external environment. And by internal environment, I'm going back to the beginning of the semester, extracellular fluid. All that fluid around your cells. The constancy of the internal osmolarity. In contrast, an osmoconformer changes its osmolarity with its environment. If we consider the extracellular fluid osmotic concentration being dependent on the environmental osmotic concentration, what do we predict for an osmoregulator and an osmoconformer? Let's think first about an osmoregulator. What's the line? Yeah, it's going to be a flat line, right? An osmoregulator is going to be maintaining constant. What about an osmoconformer? One-to-one relationship. Make sense? So we can actually plot this. And in lab this week, this is our question. Are fiddler crabs osmoregulators or osmoconformers? We're going to be having them in different environments. They are right now. They're in different environments. And we're going to measure their internal environment. When we plot different crabs, some of them are relatively isoosmotic. Arcturus, Lygia, whereas others, Artemia, are not. So this leads to our question this week for fiddler crabs. Are the fiddler crabs osmoconforming or osmoregulating? Now when we consider vertebrates, vertebrates are all osmoregulators. Fishes, amphibians, reptiles, birds, and mammals all regulate their osmolar concentration. And they do this in a variety of different strategies. So I've got a nice little handout for you here. This slide, because it's kind of busy. And I'm not going to focus on all these animals, but I do want to point out the strategies, the ways that different critters maintain their concentration. Let's start off with, you want to do freshwater fish first or marine fish?
Fresh.
Freshwater fish. Okay? Now, before you look at your paper, real simple. Freshwater fish, largemouth bass. Largemouth bass in Fort Loudon Reservoir. Or let's go to a cleaner lake. Tellico. All right. Let's go to Tellico Reservoir. Largemouth bass in Tellico. Is it going to have more solutes inside its body or in the water? Inside its body. Everybody good with that? Fresh water, right? Therefore, what's the osmotic problem? By osmosis, water is going to want to go which way? Into the fish. In other words, the fish should be exploding. Literally, without any kind of osmotic regulation, it's going to explode. Therefore, what do freshwater fish do? They constantly pee. If you've caught a fish in a lake, you know this. You catch a fish, brim, don't point the cloaca towards you. It's going to be, and it's not like it's mad at you or anything. It's doing that in the water all the time. Freshwater fish are urinating out. Freshwater teleosts are hypertonic. Another way to say it is hyperosmotic, high concentration. to their environment, and therefore they urinate out a lot of urine. They also don't drink any water. That's the other thing. Freshwater fish do not drink water. They don't have to. It's moving across their gills into their body. So they don't drink water at all. They also have to actively pump sodium into their body, or else they lose it all. So absorb salts with gills. Now let's go with it. Okay, let's go to the beach now. We just transformed ourselves. We're on the coast. We catch a red drum. Or, well, let's go with a sea bass for simplicity. Sea bass. In the water, we put that sea bass. Is the environment or the fish internal environment higher solute concentration? Environment. Therefore, this fish that's now in the ocean, what should be happening if nothing is going on in its body? It'll be losing water to the environment, shriveling up. Okay? How do they compensate for that? They drink water all the time. Saltwater fish drink water, but the problem with that is we're drinking more salt. Therefore, the gills have pumps that pump the salts out. Again, let's look at the marine teleosts right here in this line in the table. They are hypotonic or hypoosmotic to their environment. The environment has a lot more solutes in it. Therefore, they drink seawater and they secrete, they get rid of, they pump sodium outside their gills. Same thing's true for sea turtles. Sea turtles, again, they've got a real problem. They're in an environment that, and they do have to consume very salty food all the time. I'll never forget, I used to work on a sea turtle island. That's what I did in the summer when I was in college. And I would take groups of people out to watch the turtles lay the eggs, catch the eggs, take them back to them, that little protected area in the dunes. And I could predict, and it got so funny, we would be able to predict what people were going to say. Y'all know this, if you deal with the same kind of people all the time, you kind of predict when they're going to say something, when something's going to go. This never failed. We'd be up there in the dunes, all around, and somebody said, look, she's crying, she's so emotional. She's laying her eggs and her tears of joy are streaming down her face. And I would, that's actually a salt secretion, Sometimes I do that, depending on the people. I mean, they have lacrimal glands, just like we do. And they are constantly making salt, excreting it through their lacrimal glands. They drink seawater, but they pump the salt out through these lacrimal glands, tears of salt that they're always making. Just like the freshwater fish are always urinating, the sea turtles are always excreting salt. Same thing's true for seagulls. They're always crying. They're not sad. It's an excretory organ. Let's now go and talk about mammals real quick. Now, mammals have a unique problem compared to other classes of animals because mammals don't have special salt excretory organs. We can cry, but we can't cry enough to get rid of salt. Therefore, we have special adaptations in our kidney. Now, I'm going to remind you of something that I talked about several classes ago, and that is the location of the nephron in the kidney. Some nephrons are what we call cortical nephrons. This is an example. It's in the cortex. Some nephrons have a loop of Henle that goes all the way down. We call that juxtamedullary nephrons. Most animals have a mixture of cortical and juxtamedullary. Desert mammals, though, desert mice, for instance, or coyotes out in the desert. Desert mammals have all juxtamedullary nephrons. Water always being saved because of the juxtamedullary orientation. Same thing's true for whales, dolphins, dolphin mammals, porpoises. These mammals don't have excretory organs. They have to save all the water they can some way, and they do this through having juxtamedullary nephrons. One last thing I'm going to introduce you to today, and that is the most problematic of all animals that we've talked about so far. A salmon. You tell me, what's the problem with salmon we got here? Osmotic. There goes saltwater, freshwater. I've told you about, if you're a freshwater, you do this. If you're saltwater, you do this. Here's an animal that does it both. How in the world? It's a miracle. No, I can explain it to you. It has to do with the stage, the life stage. Now those spawning fish lay eggs up in freshwater streams. Those eggs hatch into what we call fry that quickly metamorphose into parr. Now these salmon parr, they live in freshwater streams up to 3 years. Little bitty salmon, freshwater parr. Whenever they are parr, they act just like freshwater fish. They're always urinating. They're also pumping sodium in their gills. So parr are pumping sodium inside their body. They then go through a process, I love this name, smoltification. Smoltification has to do with the transition as they go into the estuaries. Smoltification actually is under the influence of the stress hormone cortisol. Whenever the parr go into the estuaries, cortisol levels go up. And all of a sudden, the pumps that were pumping sodium in reverse. They're now able to change the direction of the sodium pumps. And as they move into the open ocean, the hormone prolactin, which you've heard of before because you've heard of it in mammal milk production, in a salmon, that allows them to have maximal secretion of sodium at the gills. As long as prolactin levels are high, they're always getting rid of sodium. And this happens until they begin their spawning, when the prolactin cuts off, and it goes the other way. Real simple. Somebody summarize to me, if you're a salmon, what's the big difference in your freshwater life and your saltwater life? Sodium pumps and the direction they just pumping on gills. Pretty simple. It's all. And that's what allows these. By the way, this is called a diadromous animal. There are lots of them. Eels are diadromous. Some crabs are diadromous. They can go from one salt environment to the other.
Is that a bull shark?
What's that?
What about bull shark?
I don't have any idea. Can a bull shark go to freshwater?
That's like, I think, I don't know. That's, they made a book.
Yeah, estuary, but that would be like 50%.
Yeah.
All right, tomorrow in lab, we are picking up on this whole experimental idea of osmoregulator, osmoconformer, and Thursday, one of the most difficult lectures I'll have to deliver to you. It's on pH homeostasis.
So Thursday's the day to skip.
It's the one where you get your money out of it. I mean, this is what you pay me for. It's these difficult lectures on PHME Station.
I don't know. You've told me to drink wine every day, and now you're giving me experiments to do with drinking. So it's like, I feel like I've got my money's worth.
It's probably over the other side. Dr. Crane,
Yes, ma'am.
did you say that natriuretic peptide decrease or increase?
Should I? Yeah, should go down.
Okay.